The Journal Club this week is going to focus down on a paper from 1939. And our last Journal Club was from 1905. So you might want to wonder about what happened to the first essentially half of the 20th century. And essentially what was going on was that there were a lot of experiments being done where different kinds of things were injected into experimental animals. And then serum was taken from these animals, and different assays were developed. I think I've mentioned that before, to see whether they could find antibodies in the serum. And so the immune response during this half century was mapped by measuring antibodies after the injection of the antigen. So this was really at a whole person or a whole animal level to try to understand how the immune system was working. But the overriding question that people wanted to know about after they discovered antibodies in the first place was, so what are these things, these antibodies? What are they comprised of? And how do they work? How do they do whatever they're doing? It was clear that they reacted with an antigen because they could see it reacting under the microscope with precipitations were formed and agglutinations and so forth. But they didn't know how that was all working, and they knew that they needed to figure this out. And the other thing that was sort of at the back of everybody's mind was, what's the difference between a successful immunization and anaphylaxis, so that the patient died? You wouldn't want that to happen. And so that's a conundrum that was going on. And in 1924, a classic paper was published by two investigators from the Institut Pasteur, Kabat and Guerin. And they listened to Pasteur and they said, okay, we've got to, in order to get a vaccine for tuberculosis, we have to attenuate the virulent organisms. You know, it was responsible for so much of the death and disability and so forth and morbidity during particularly the 19th century. So they listened to Pasteur and they passaged some tuberculosis organisms that they isolated from cows, bovine TB, and they passaged it and passaged it and passaged it and passaged it. And it was interrupted. They started in 1905 or so, and but all of their experiments were sort of interrupted by World War I, and they had to stop doing that. And so they took off after that and kept on passaging and then testing to see whether or not experimental animals would get the disease or they wouldn't get the disease. And ultimately, they came up with a culture that ultimately became called BCG, Bacillus Kabat Guerin. And that vaccine, it worked and it was adopted in Europe, but there was all kinds of controversy associated with it because the efficacy, and now we've come in the COVID generation, everybody's worried about or not worried, but concerned about how efficacious their vaccine might be that they get, whether it's 95% like the first two that were approved by the FDA here in the United States, or whether or not it was less than 95%, which is really a very good vaccine, by the way. Anyway, the different reports were coming out saying that it didn't work at all, or it worked up to 80%, 85%. And in the United States, they never really adopted BCG vaccination, whereas in Europe, it is still being used, and kids are immunized with BCG, whereas over here, because the disease was not as prevalent as it was in Europe, they decided not to do it for all kinds of reasons and so forth. So that's BCG. It's on the website. 1924, Kabat and Guerin. It's all in French is the problem, and I haven't read this paper, even though I can quasi-read French. I haven't read this paper in its entirety, but you can go online and just Google Kabat and Guerin, and you can find out the history of this whole business. So that was 1924. Then in 1931, there was another vaccine that was reported, this time for yellow fever. And yellow fever, in a nutshell, what yellow fevers do is due to a virus. Of course, we didn't know what viruses were back in 1931. or 1937, I'm sorry. But yellow fever is due to a virus that causes a severe hepatitis. That's why they call it yellow fever, because the patients turn yellow, and it was lethal. It was a tropical disease primarily, but in the summertime, particularly on the East Coast, it would work its way up to as far north as Washington, D.C. And everybody just left the city in the summertime. They went to the boondocks, up into the mountains for the good, clean air and so forth and so on. Well, it was found that this disease was being transmitted finally around the turn of the century by a mosquito. And so after that, the virus was identified and so forth. And so a vaccine was something that everybody wanted to have. And again, methods were devised to try to attenuate the virus. And there was the paper that's on the website describes the background on this, is that they passaged this virus. And ultimately there was a random mutation so that it was lethal, and all of a sudden it wasn't lethal anymore. And of course, this is before genetics, really, or molecular genetics. This is before we knew about genes and DNA and all this other stuff. So that's sort of the source of the word variant. We're inundated with variants of coronavirus, and I've sort of taken a brundage to that, because I said to myself, what are we still talking about variants for? We should be talking about mutants, and people should learn about what a mutation is and how DNA works and how the genetic code works and so forth and so on. But we're still not there, and this is now 2021. Maybe we'll get there by 3021. But anyway, the paper that's on the website from 1937 is about the yellow fever vaccine. And that was the only vaccine between the time that Pasteur said that he was going to prevent all contagious diseases and with prophylactic vaccination and or therapeutic vaccination like he gave for rabies, and we could cure diseases. And so the problem was that what he told everybody we needed to do is to attenuate these bugs. But that would prove to be a little bit difficult. And actually, when Pasteur thought he had attenuated the bugs that he was using for chicken cholera and anthrax, he really didn't attenuate them. But that's another story, and we'll get into that later, perhaps. Anyway, the paper that we're going to look at now is from 1939, and it's published in the Journal of Experimental Medicine in 1939, and it's from the biophysics department in Uppsala, Sweden. And the authors of this paper are Arne Tiselius and Elvin Kabat. And this is an interesting story. The title of the paper is An Electrophoretic Study of Immune Sera and Purified Antibody Preparations. And the reason that this is a very important paper, because of the fact that it really gave, for the first time, a means to identify and characterize biochemically what antibodies actually were. And what electrophoresis is what you take a solution of protein that you want to characterize, and you apply an electric current to that. And the different proteins will migrate, positive or negative. I was never good in electricity in school, but anyway, the proteins will migrate dependent upon their charge. And so different proteins have different charges and they migrate differently. The other way you can separate proteins, and that had been just started to be able to do that also in Uppsala, in Sweden, was the ultracentrifuge. And you could separate proteins, therefore, by their size, their molecular size. Svedberg was the primary investigator in the ultracentrifugation. Kabat, to give you some background, his parents were from Eastern Europe. They immigrated in the latter part of the 19th century, and they ended up in New York City. And when they came through Ellis Island and whatever, they got rid of their name was Kabatschnik. They got rid of the schnick, and so they just ended up being Kabat. Kabat, I think we pronounce it these days. And so it's a very typical American story about this time where we preferentially let in immigrants, particularly from Europe, who were so-called Caucasians, meaning that they had white skin. And Elvin Kabat, the co-author of this paper, so Tiselius was the leader of the lab, and Kabat was a postdoc. And his history and his background is that he graduated. He went to the City University of New York, and back in those days, CUNY, the City University of New York, was really prestigious, particularly among the immigrant population in New York City. And it was very, very prestigious to go there and to graduate. Well, Elvin graduated from the university there. He went there at the age of 15. He graduated in 3 years at the age of 18 in chemistry. So this guy was a star. And then he went and he worked with one of the major stars in immunology, Michael Heidelberger, who was at Columbia University uptown. CUNY was downtown. And within 4 years, he got his PhD with Heidelberger. And that left him, put him about 1936. And then then of course, this paper we're going to talk about is from 1939. Kabat went to do a postdoc in Sweden, in Uppsala, at this biochemistry department with Tiselius and Svedberg and the rest of them. And these people were really luminaries. And they were really at the cutting edge of this whole new field of characterizing proteins and that sort of thing. He was only there a year, say 36, 37, 38. Of course, we all know what happened, which is probably one of the reasons he wasn't there for very long. But while he was there, he really, and of course, he was 22 in 1936. So he was just a kid, you know. But this paper made his career. And from there on after, for the next 70 years, he was Mr. Immunochemist. This was the beginning of immunochemistry and the characterization, which was focused all about what the characterizations of antibodies was all about. At the time, it was already known via this electrophoresis business of serum that there were 4 peaks that you could ascertain on this by separating the molecules on the basis of their charge. And the fastest moving in the electrophoretic current was albumin. And then there were globulins, which is a word that I had to look up even now to find out exactly, so what are you talking about? And what I found out was that all albumin was globular, so it was round. And then but it's the fastest moving. And then there is alpha. Without the Greek lettering, what would science do, you know, whatever. So there was alpha, beta, and gamma globulins. And of course, now gamma globulin just rolls off your tongue because everybody knows that that's where antibodies migrate in this electric current. And so the slowest of the globulins is alpha and then beta, the fastest is alpha and then beta, and then gamma, the last one, is the slowest migrating kind of globulin. So that was known. Now the question then was, where are antibodies in this whole thing? And Heidelberger, back in New York, he sent over serum from hyperimmunized animals, horses and rabbits, goats, guinea pigs, and all kinds of things, so that Kabat could do his experiments in Tiselius's lab, with Tiselius is overlooking and making sure he's doing everything properly and so forth. Rabbits particularly were really good. Rabbits made huge amounts of antibodies, by the way. And so they've been a favorite of the immunologists ever since, you know, the beginning. And so one serum in particular was raised against egg whites, egg albumin, egg whites. And that became a favorite antigen to shtup into horses and goats and rabbits and so forth and so on. And it had been known that the antibody activity migrated in the gamma globulin portion of the electric field, electrophoretic apparatus. And therefore, the purpose of the paper and the purpose of the experiments that Kabat was wanted to do was to compare the electrochemical properties of the antibody in serum with purified antibody under the same electrophoretic field that he was going to do. So that's what they did. And now I want to move to share my screen with you, because there's several tables in this paper. Maybe I should preference what I'm going to say is that between 1900 and 1970, really, the major way the data, so that was the time when scientific papers started changing. So that back in the 19th century, they, like Bester would stand up and hold forth and tell everybody in the audience, well, I did this and I did that, and this is what happened. But data were never published in papers, results, methods. They just had to take Pasteur's word for it that he was a genius, which he told them very well. And so then there's the composition of a scientific paper became established, so that there was an introduction, there were materials and methods, there were results, there were discussion, and then there was a conclusion. The results of this paper were detailed in several different tables. And those tables I found, reading these kinds of papers over the years, are very difficult to figure out whether or not whatever they're saying in the text is really true from what the data are that are in the tables. But figures were much, much better. You could just glance at a figure and you knew exactly what was going on, whether or not they're telling the truth or not in their discussion or whatever. The essence of this paper is all in figure 1. And so this is what I want to share with you by the screen sharing. There we are. Now, this is figure 1A at the top and figure 1B at the bottom. And I want to draw your attention to the 1B at the bottom first, because on the left here, we have a very sharp peak. That's albumin. And so this is an electrophoretic diagram. And then we have alpha, and then we have beta, and then we have gamma globulins in this plot here. And you can, from this, you can discern that there's a lot of albumin and not as much alpha, beta, and gamma globulin. Here is that same sera up here in the A, what it looked like before the antibody was removed. So the gamma globulin fraction was sky high. And most of that protein that is in this peak is antibody to the egg white. And this was from a rabbit serum. And it basically, what this did is that from there on after, in the clinic and wherever else, these kinds of profiles were published and shown and used in the clinic and used in medicine, because you can tell the relative concentrations by calculating the area under the curve. Each of these peaks, you can get the relative proportions of albumin to gamma globulin and so forth. So the conclusions of this paper, and I'll go back now, I'll stop the screen share. So the conclusions of this paper are very simple, and I've left a couple out actually, but the horse antibody migrated as a new serum peak between the beta and the gamma peaks. So there was an extra peak in there, and that was the horse antibody. The rabbit antibody was just superimposed on the gamma peak. And you could use, and they showed in this paper, that you could use electrophoresis to separate the antibody from the rest of the proteins, the rest of the serum proteins. And you could use you could precipitate the antibody out by throwing in antigen. And then you would get antigen-antibody complexes, and you could separate them on the electrophoretic field, and then you could purify these things that way. And as I said, you could use the electrophoresis to determine the concentrations of these things. And so for the very first time, together with the ultracentrifuge that you may have heard of, 7S and 19S immunoglobulins, and then the S is for the Svedberg constant, which tells you where these proteins would migrate when a tremendous centrifugal force was applied to them. So you had two different means to characterize antibodies in addition to their antibody activity for the very first time. And I will stop there. This paper is only half a dozen pages long. I recommend everybody read it, and then we'll go on from there. So this is 1939, and then of course we had to fight wars, and we had a World War to come, and so there was a hiatus of scientific reports during the war years, and thank God there were only 4 or 5 of them for most of us. So I'll stop there. Thank you. So if you've enjoyed this video, please like, subscribe, and sign up for my newsletter, where I'm serializing my new book, which is called The Quest for New Knowledge. You'll find a sign-up link below. Hey, thanks again. It's been great.